You guys, let's go ahead and get started.
That's where we teach.
I'm Terry Davis. I am professor of medicine and pediatrics and in the Feist-Weiller Cancer Center, and I have these cool guests to help me teach what I think is going to be a very exciting Grand Rounds. Dr. Glenn Mills is head of medicine, head of the Cancer Center, and head of genomics and precision medicine at LSU. And Ellen Friday was, for the medical students, she won the award as best clinical teacher last year. And she is the technical director of the biobank here, the cancer biobank, and genomics lab. And Mindie Kavanaugh used to sit where you guys are sitting. She was a medical student resident here. She did her fellowship here, and now she's an oncologist on the faculty. And I've asked her to come to see how she applies all of this with her patients and with herself. Come in, Sandy. Are you in the right place? So I was thinking about this Grand Rounds, and I think there is so much happening in medicine now. It is just exploding. And what's exciting when I look at it, this audience, you guys are here just on the cusp of this. This thing is just about to take off based on all the genomics work we've done in the last 20 years and all the technology that's happened. So, precision medicine, genomics, biobanking, moonshots, 21st century cures, what's all of this? Is there any of this happening at LSU or at the VA? Why is it important for students or residents or faculty? Why is it important for our patients or the public? I've invited Christy Gustafson, who's the new head of the Community Foundation, in my attempt to educate the public here. How will physicians and researchers communicate these things to the patient and the public? What are the legislative funding and policy initiatives driving all of this? And why is it timely? So in 2015, President Obama announced the Precision Medicine Initiative. It was a bold new model of research using big data to improve our understanding and treatment of cancers first, and then other rare diseases. They are calling this, all of us, to kind of be more conversational. It is a $1.4 billion NIH-funded research program that's going to build the largest, richest, most diverse data set ever to provide a research platform to improve health and treatment of disease. In Europe, they're doing similar things. I don't think they have so much boldness going on, though. The national rollout is this spring. They're going to enroll 1 million U.S. adults to give consent for extensive characterization of their biological specimens, including RNA and DNA and whole genomic sequencing, if cost permits, all linked to their electronic health record. And then researchers from all kinds of organizations will have access to this. So Glenn, do you know how much it costs now to sequence your whole genome?
Probably you're looking, if you do it just by itself, probably about $1,800. But the way we do it, where we run multiple samples, probably about $1,400.
So Francis Collins, who's head of the NIH and a UNC graduate and a folk musician, bluegrass musician. Also, he's unusual in the sense he's head of the NIH. He was a leader in the Human Genome Project. But he was NIH director of both Obama and Trump. And I saw an interview with him, and when he went to be interviewed by Trump, he rode the bus from D.C. to Trump Tower. He was probably the only one who didn't have a corporate jet going up there. But anyway, he says the concept of precision medicine isn't new. What's new is the dramatic improvement in our capacity to do this broadly. With our advances in molecular biology, genomics, and bioinformatics, we have powerful methods for characterizing patients, and we now can have large-scale biological databases. So the first out of the gate is going to be cancer. Longer term, other diseases, including brain disease and drug addiction. It also taps in on cultural trends of increased connectivity and Americans' growing desire to be active partners in medical care and research. PCORI and other national funders now want community advisory boards. They want patients and the public involved in research from the get-go. They're also interested in how we disseminate findings, not just in medical conferences and scientific journals, but how do we get the word out? How do we keep medicine from just being on the bookshelf to being applied widely? So, PMI is not a clinical trial focused on a specific medical problem, but a national initiative to develop ways to measure all kinds of diseases based on environmental exposure, genetics, and their interaction. They're looking at causes of individual differences in response to commonly used drugs. Why do some drugs work for some of your patients and not others? Discover biomarkers that increase or decrease the risk of common diseases. The beta was launched in 50 sites in June, and I think there were 46 physician practices health systems involved, but by October, they realized that the messages need to be simpler. The public didn't understand what they were doing. Also, ethics hadn't caught up with the technology, so privacy, trust, transparency, participant preference, all of that hadn't been worked out, even though we now have the technology to do a lot of these things. So what are you asked to do? Well, if you're over 18, you can sign up. Children are going to be enrolled later. You give your name and address. You don't have to give your Social Security number. I guess they realize that patients like LSU patients don't like to give their Social Security number. You give blood or urine. You fill out all kinds of surveys about your health, home, work, environment. You go to a site and you're weighed, and your height, weight, blood pressure, heart rate, waist and hip circumference are all taken. You allow them access to your electronic health record, and you're paying $25 if you're measured and give a sample. So the veterans have a program too, and they're going to try to enroll 1 million veterans in the next 5 to 7 years. What's different about theirs is they're adding military exposure. And Overton Brooks is the site for their 1 million vet campaign. And researchers will study diseases like diabetes and cancer, but military-related diseases such as post-traumatic stress disorder. So I thought that was pretty cool that our VA is part of that. Have y'all heard anything about that, Glenn, Mindie? No. Overton Brooks? Okay. Well, there you go. So also, Connie Arnold, my colleague, and I, we got a call from UAB, and I just thought I'd let you know this. Sometimes it frustrates me that Louisiana is not Alabama and LSU is not UAB as far as health centers, but the legislature in Alabama gave them $2 million, UAB, to have a precision medicine initiative in Alabama where they're going to enroll 10,000 adults in Alabama from each county in the state, and they're all going to give a small sample of blood. What they think they're going to do is they're not giving them $25, but they're all going to provide all of them with a 23andMe. Now, I don't know how much that cost, or who's going to explain it to you. I don't know what needs to be explained. And then they want to educate providers across the state so they can talk to their patients to have a genomic-ready population and workforce. And the bottom bullet is the bottom line, I think. The initiative, according to their website, funded by the state, could be transformational for Alabama, improving health care and stimulating economic development. So you're going to find out we're doing some of that stuff here, right here in River City. So what is the 21st Century Cures Act? Well, right before the Obamas left town in December of 2016, Congress signed $6.3 billion legislation to speed up things. Development of medical products and research advances to patients faster and more efficiently. And so there was a lot of money for cancer, there's a lot of money for brain, there's a lot of money for drug prevention, and there's also money to support state efforts to contain the opioid abuse and improve mental health services. And the legislation authorizes NIH to finance high-risk, high-reward research. Now, there's a lot of implications for the FDA. I don't know if drugs are going to be rushed to market or what the implications for this are. So here's some provisions for this. The NIH will, Glenn, cut bureaucracy, require data sharing, even with South Louisiana and North Louisiana, provide privacy protections, and here's what applies to some of y'all in this audience. Support new researchers and assist in student loan repayment, and they want to encourage diversity, allow Americans of all stripes to participate and benefit from NIH-funded studies. So what about the Moonshot? These guys are more involved with that, and one of the blue ribbon panel members is head of the Cancer Center in New Orleans, Augusto Ochoa. So they want to double the rate of progress against cancer. Advance cancer research, basic science to advanced clinical trials promoting new therapies. They want to establish a cancer immunology network to learn more about why some therapies are effective with some patients and not others. Accelerate our understanding not only of cancer treatment, but prevention, early detection, and care, and improve patient access to state-of-the-art care and research. One of the other things that's expanding with this is more people are living longer having benefited from the cancer treatment is what are we going to do about all this survivorship? So here's the challenge. Why cancer first? Because in the aggregate, it's one of the leading causes of death worldwide, and the incidence is increasing as the population is getting older, and rapid developments in genomics has created novel opportunities for patients to participate in clinical trials offering genomic-targeted therapies. You'll find out more about that in a few minutes. But less than 5% of U.S. adults with cancer are currently enrolled in clinical trials. Steps are needed to ensure all Americans, including those with limited resources, those living far from major cancer centers, have access to leading treatment, prevention, screening, research, etc. So just to be clear, I know y'all get this drilled into your head all the time, but a clinical trial is a research study involving humans, and they're usually randomly assigned to 1 or 2 or 3 treatments. And in the past, what you're looking at is the biological outcomes. Is the A1C better or whatever? Now, what's new in this new NIH definition is they're looking at health behavior and quality of life. And this survivorship is all about behavior and quality of life. And there's a strong recommendation to have a central IRB. Well, Glenn can maybe say something there. So part of the problem, and one of my and Connie's interest, was the disparities in clinical trials. The disparities exist in recruitment, trust in clinical trials, and biorepositories among individuals who are low income, have limited literacy, racial and ethnic minority groups, or who live in rural areas. And a lot of our patients live in rural areas. Currently, less than 18% of people on clinical trials for anything are minorities. So why is that? Diverse participation is needed to help identify how specific drugs and treatments are most useful for different groups and different patients. If patients from diverse groups don't participate, knowledge of best practices will remain skewed towards white patients and therapies that work for them. Up until recently, most clinical research and a lot of drugs and a lot of therapies were developed on research on either coast with white folks, particularly white men. And that's why the NIH now says, you got women, you got minorities, who you got in this trial? So here's what the literature says. The barriers to minority participation are misconceptions about research. Am I going to be a guinea pig? They don't see the benefit for people of color. They are afraid of being discriminated against. The potential for side effects, inconvenience, cost. Facilitators, they need adequate information. Well, where are they going to get this information? Community education at African American fraternities and sororities can increase awareness. Partnering with trusted community figures, may help overcome barriers and make it more accessible and acceptable. Another perk for anybody about being in a clinical trial is free medical services, better medicines, contribution to future generation, particularly if it's non-invasive or if there's money involved. But we need to make sure that that these clinical trials, not only that people have that information from a trusted source, but it's easier for them to participate. So here's some communication challenges. So the cartoon says, perhaps you'd like to donate some of your genes to a biobank. And she says, what's a biobank? What am I agreeing to? So I think in some ways, many patients might be thinking this. So what's happening is there's all this precision medicine, genomic testing initiatives are creating a widening gap, not just between the researchers and the patients and the public, but between researchers and primary care docs. Clear, accessible information on clinical trials, genomics, and biobanking is lacking. What are understandable and acceptable message? People need to be clear what they're consenting to. Data validity. If you're going to fill out all these surveys, do they understand the survey? Are they clear about how to answer it? How valid is that data? And we need, I think, to evaluate creative use of automation to deliver easy to understand, not scary clinical trial information, consent, and HIPAA forms. Right now, we're doing this on the backs of people that maybe take them an hour to consent somebody for a clinical trial, and Mindie and Glenn can say more about that. So this cartoon kind of sums it up. For me, one of the genomic experts at LSU New Orleans I work with is sort of like this guy with his hand out here, and little girls are asking, what are little girls made of? I thought he was going to say sugar and spice, and he got into the weeds about the human genome and all of that. And sometimes that's part of the mismatch in communication. So I recently, where's Jay Marion? Recently heard Alan Alda. I don't know if medical students, are y'all too young to know MASH? Did you ever watch MASH? Hawkeye? It was a cool show. I loved it. Anyway, what Alan Alda is now doing after he left MASH, he's running the Center for Communication Science at Stony Brook, and his job is to teach researchers and clinicians how to talk to the public, how to communicate clearly. And here's some of his pearls. Communication does not take place because we tell somebody something. Think about talking to your patients this afternoon. Communication takes place when we observe patients closely and get signals that we're on the right track, that we've started at the right place, a place they're familiar with, and track their ability to follow us and confirm their understanding. I thought this was very insightful. We forget what it's like not to have certain knowledge. You guys have learned so much, and we forget what it's like not to know these things. We need to describe things in ways patients can understand and be interested in. You don't want to bore them. If people are bored, they stop listening. And we must keep patients with us so they can take in what we're trying to teach them. And I love this too. In communicating about research, clarity is not dumbing it down. Science is never hurt by too much clarity. So, Connie and I wanted to know, what does the public know about precision medicine, genomics, biobanking? What information do they want? How do they want to receive it? What do primary care doctors know? What do they want to know? How do they want to learn about clinical trials at LSU? How do clinicians and researchers explain these in a way that's useful? And how do we make the information and participation easier to access? So we did focus groups of safety net patients and providers and adults and predominantly African American social and church groups. We're going to find out what were they aware of? What do they understand? Trust, acceptance in clinical trials, and help with how to express this. So we did 14 focus groups in 4 parishes, and of the 78 patients, 78% were African American. We had 25 providers. We talked to community clinic patients and providers, Feist-Weiller cancer patients and their clinical RAs, LSU medicine residents, Council on Aging, African American sororities, African American churches, and Alzheimer and Parkinson's support groups. So here's what we found out. Much to my shock, people were more aware of clinical trials and personalized medicine because of everything on TV. They knew about 23andMe, and they had seen, Glenn, all those cancer ads saying if you went to these cancer centers, you could get cured or live another day for your grandchild. Almost all participants were interested in research but not aware of studies or how to find out about them or enroll in them. They were particularly interested in learning about studies focused on diseases that they or their family had. They said, don't call it, they were interested to give back even if it wouldn't benefit them directly with biobanking. Confidentiality, however, was a concern. Why do you want to take my blood? What are you going to do with it? Rural patients wanted to know where the bank was. It's on the 6th floor, right sort of across from my office, you'll see. How much information about me will be kept? What will it be used for? Who's going to be mucking with it? And will I get my results back? Right now, with biobanking, you don't get your results back. So here's their attitudes. African Americans said, you don't need to target African Americans. The message needs to be all people are needed for studies to improve treatment and find cures. Researchers shouldn't use the rationale that blacks have been underrepresented and most treatments are based on whites. Targeting blacks made them suspicious. Cancer patients, Alzheimer and Parkinson caregivers were highly interested in clinical trials. Few, however, without a diagnosis of cancer had ever been offered participation. Here's what they said you should call it, a study. Clinical trial is jargon, it's inside language. Biobanking, genomics sound intimidating and alarming. These things didn't mean anything to the public. Wording suggested your blood or tissue are going to be stored in a bank. And don't get in the weeds about genomics, as the cartoon implied. So what did the residents and other primary care docs say? They were all interested in clinical trials and wanted closer ties with researchers. The community docs suggested on-site in-service and telemedicine conferences that give specific information about clinical trials. They wanted to be more involved in studies, wanted researchers to give them up-to-date information about the trials. Maybe Sanders Hearn could weigh in on this. How do you want to learn it? They said email, US mail, or text. It's hard. They are so busy, it's hard to figure out how you inform them. Medicine residents wanted grand rounds and noon conferences. Some residents said the electronic health record could flag patients who might be candidates for trials. Others said, no more pop-ups in the electronic health record. Too many now. So what are their barriers? Lack of time to identify trials and explain them to patients. And some LSU residents said, LSU patients have a hard time just getting here for a medical appointment, much less a research appointment. The facilitators, they wanted information. They wanted to be involved. They wanted plain language handout and a card they could give patients to call if they wanted more information. Also, primary care docs were asked to give talks in the community. Learning about these kind of things will give them something to talk about when they go to the Rotary Club. So, here's what the LSU resident said. It's critical for physicians to be familiar with the study before they talk to patients. What is their patient committing to? Patients want to know, how is this going to affect me? What's the time commitment? How often do I have to come up here? What are they going to do to me? The physician has to understand patients' pre-existing belief system. What are they already to believe? And I loved it. Our medicine resident said, I tell patients this is going to help us better understand the disease you have. You have benefited from research done in the past. CRAs tell patients nothing happens in cancer treatment that isn't the result of prior research studies. Most people have participated before you to get us to where we are in cancer treatment that you're currently offered. So here was the research associate's perception. Cancer patients are usually receptive to clinical trials. They'll do whatever Mindie and Glenn and the rest of the oncologists suggest. They also said that the relationship with the coordinator is, Eileen, important. Participation decision is also dependent on what's most important to the patient that day. They like to feel like they're important, they're a part of something bigger, knowing that there are thousands of people participating. They like the idea of helping somebody else. However, the CRA said that they thought they had noticed a difference with highly educated cancer patients becoming more resistant to biobanking. They don't want research done on their tissue or their mother's tissue of blood if they don't know what it's going to be used for, who has access to it. So here are the recruitment recommendations. Information on clinical trial needs to come from a trusted physician. Nobody suggested the internet for our patients or most of the public here. Patients suggested putting posters in waiting rooms to prompt them to ask the physician about it. If the clinical trial information was on TV or newspapers, they'd still ask their doctor. Community groups suggested speakers at their meetings or trusted publications. I asked, what's a trusted publications? AARP, Council on Aging, or Willis-Knighton or Schumpert newsletters. I notice y'all are promoting this in your newsletter, Genomics. And some participants suggested a health fair. So in concluding my part of this, before we go on to my guest, African American and white adults were interested in clinical trials but not clear how to learn about them. Most were willing to participate, but they'd never been asked. Participation of vulnerable patients in clinical trials and biobanking could be enhanced by developing working relationships with community physicians. A community physician is a conduit to their patient population. Structured methods for closer interaction with community docs needs to be established collaboratively. And participation in biobanking with underserved and rural communities could be enhanced using the Feist-Weiller van. So now I want Glenn Mills to tell you what is happening at LSU. Let me just ask you before he starts, how many of you knew we had a Center for Genomic and Precision Medicine? Y'all know that? A few faculty? Okay. Well, what's happening, Dr. Mills?
Thank you. Thought we would do a couple of things. We're going to have Ellen talk about our genomic testing facility and our biobank. And Mindie is going to discuss how the results of genomic sequencing and testing are affecting her patients in the clinic. The precision medicine is a key word nowadays. And as you see, there's a lot of money in this being thrown out there by the feds and by other agencies for research. UAB just got $2 million. Ochsner just got $1.3 million from a thankful patient to begin a Center for Precision Medicine at Ochsner. I was down there talking to them Wednesday about their new center. So we've established within the Cancer Center a Center for Genomic and Precision Medicine, and we hope to establish this, and it's going to be established as a campus-wide effort, not just in cancer, but in brain diseases and in cardiovascular diseases, because genomics has implications across the board. Cancer has just sort of led the way because I think there was so much money spent by the NCI in particular into looking at the genomics of cancers, then that's sort of why it's led the way. But if you think about it, many of our diseases besides cancer are genomic based. We've known this for decades, okay, with our family histories telling us we've got these, histories of families with early onset Alzheimer's. Well, why is that? It's probably genomic based. There are probably changes in our genes that do this. So we thought about 8 years ago now, 7 years, invested in bringing genomic sequencing to the campus, where the Cancer Center brought the first sequencer here, and we began to encourage our faculty to become actively interested in research with genomic sequencing by underwriting the costs of running the sequencer. We now have 3 sequencers here, and we stay pretty busy. There's 2 in our core lab right now, and then 1 in our genomics testing facility. We are a CLIA-approved testing site, so we are now offering commercially genomic sequencing for cancer patients. Just began in the last month, and Ellen's going to tell you a little bit about that. We've run about 30 cases so far, doing about 6 cases per week. And we're going out trying to drum up business all across the state and hope to be doing the sequencing for LSU New Orleans, for Ochsner, and for others across the state so that we can begin to develop a very large research database. We're going to be participating in the All of Us. We're going to be offering that once they open up the opportunity for other sites to come on board. I think that's going to be a very important thing for us to do here on the campus, is to recruit patients in for this. So we're committed to helping sponsor that in the Cancer Center, and I know the other centers are very interested in this as well. But Ellen, why don't you tell them about biobanking?
Let me give Ellen a couple of words of introduction as the technical director. Now, I read what students had written about her in the past, and one of the things that I don't want to oversell you, but that she's more interested in your learning than in teaching. So hopefully we will all learn something. Also, I naively didn't know we had a biobank when I was doing these focus groups, and then I found out it was right in my face every day. So here it is on the 6th floor. We all pass it every single day, so we all know where it is. So Ellen, tell them what's happening, Miss Award-winning teacher.
Just a little bit of a background of sort of what biospecimens and biobanking is.
You can come up here if you want to, since you turned in slides. I was just doing this one. Yeah.
So basically a biospecimen is anything that you take from the body, tissue, blood, plasma, urine, used for diagnosis, analysis. These specimens can be not just the specimens themselves, but the components that you get out of them, like the DNA, the RNA, and the proteins. We are the biobank. We are the library. We keep these specimens. We are their guardians. We get these specimens, and we're their protectors. We're not only the protectors of the specimens themselves, but we're the protectors of the patient's information. Because the specimen by itself is useless. It has to be properly annotated. You have to have the proper patient information with it. A specimen without any information about where it was taken from or anything about the patient is essentially a useless specimen. What do? We have a very long name. We are called the Feist-Weiller Cancer Center Tissue Serum Repository, or we call ourselves the TSR for short. What we do is we go and we obtain informed consent from the patient. So any specimen you get from us has been properly documented and consented. We collect these specimens. If there is any processing that has to be done, we process them, we store them. We store them under controlled conditions. We maintain a protected database. Again, we're protecting the patient information again, the patients who have concerns about this. We have a protected database that have the specimen information and the patient information. We perform quality control, and what that means is, periodically I'll go dig back in our freezer, pull out a random specimen, make sure that it sits what it's supposed to be, that we properly documented it and stored it, and that if you're looking to get DNA, RNA, or protein from it, that it's been stored under the conditions so that that's still possible. We also offer some services. For those of you who are clinicians and you want to do projects and you don't have a research lab and you don't have a collaboration with a basic researcher, we will do some things for you. We will do some immunohistochemistry, histology work for you for a small fee. We do DNA, RNA isolation services for you. You may wonder what we collect. We collect fresh tissue. So we will collect this at the time of frozen section or when it's down in the gross room from prep. Now, what you have to realize is all this is taken after pathology has verified that they have sufficient material to do patient diagnostics. So the patient care always comes first. And we hold these samples in quarantine for an amount of time. So if the pathologist says, we need, John Smith's kidney specimen from last Tuesday, we give it back. Patient care comes first. We collect a formalin-fixed tissue collection. We collect blood products. So on the day of surgery, we'll ask the patient, will you consent to have an extra vial of blood drawn? We process that. We store the plasma. We store the serum. We store the Buffy coats. We store bone marrow aspirates. So all of these are available for any research projects you may have. You just have to have an IRB-approved protocol. You come to the repository, fill out our request form, and we will provide these samples to you. As of right now, we are providing these samples free of charge. So it's not an economic burden on you. If you had to go out to buy these from other places, I was actually doing some comparable pricing today. There's a company that for 10 million peripheral blood mononuclear cells from a newly diagnosed CML patient, they want $3,000. So we're a good bargain. So you should come see, and you should come talk to us, because I go back to the words of Bill Grizzle at UAB, who's sort of one of the fathers of biobanking. A specimen that's been in my freezer is a useless specimen. If somebody out there isn't using it, I might as well have not collected it. So the other hat that I sort of wear for Dr. Mills is the new Genomics Laboratory, what we're really excited about. We are located in the BRI building on the second floor, and we offer clinically relevant testing for solid tumors. We run a panel called CancerPlex. CancerPlex is a panel that has 435 genes currently. These genes are things that can be targets of FDA-approved therapies, maybe therapies in clinical trials. They offer such things as DNA repair genes, cancer susceptibility. We do microsatellite instability. We do tumor mutation burden. For those of you who work in cervical and head and neck, we do the HPV 18, EBV. All of these are available on this panel. What we do is we have an order form. You fax the order form to us for your patient. We then take over and do the work. We'll request the block from whatever pathology lab has it. Catherine Chadois, who's our pathologist on our study, she'll come in, she evaluates it, marks the sample. We extract the DNA. We do an enrichment process and all the library preparation that's necessary for sequencing. We sequence it, and then we transmit the data to an outside partner who does our bioinformatics for us. We partner with a company in Boston called Q. And in 7 to 10 days from when we receive the specimen in our laboratory, we return a report like this to the physician. Now, this is just the front page. Our reports are an average of about 3 to 5 pages that we strive to make clinically relevant. We know you're busy. We know you don't want to have to sift through 10, 12, 14, 15 pages. We strive to make it very relevant for you. It will have things on here it will show, such as, did this patient have a mutation? Is there an FDA-approved drug for this mutation? Is there maybe a drug not in that particular patient's tumor, but maybe in another disease indication? It will also point you toward maybe a clinical trial that this patient might be relative for. Not only do we show you what's positive in the patient, what's changed, we also demonstrate here on this front page the pertinent negatives, things that are also relative. It's just as important to know whether a patient is negative for a mutation as it is whether it is a positive. We show you the microsatellite instability, the viral status, the tumor mutation burden, and then finally at the end, we just give you a small snapshot summary that's very personalized towards your patient's results and their specific disease. We're excited to offer this, as Dr. Mills said. I went down and checked this morning. We're actually doing the prep on patient number 34 this morning. So we have returned, as of last week, we have returned, out of these 34 patients, we've returned 25 curated reports to our physicians. And we're excited to see how this is going to impact treatment. If you have questions about either the genomic sequencing or the biorepository, please don't hesitate to contact me. My email, you can contact me by email. It's under rfrida@lsuhsc.edu, and I'll be happy to answer your questions. I'll write it up here on the board before we leave in case you're interested. And I think at this point, I may just turn this over back over to Terry.
Let me make a couple of comments. I don't know if it's very modest. A word about the biorepository. It's just not a cancer biorepository. We take samples from anybody, and we've got many non-cancer using this, right? Because it's an IRB-approved repository. It's got all the safety features you have to have, all the bells and whistles in the software to keep data confidential and to keep HIPAA information hidden. And it's a huge investment that we've made, and we want everybody from the campus to participate in this. And that's why we're doing things free of charge right now, putting the tissue away. The surgeons, we've had the cardiovascular surgeons and the vascular surgeons put samples away. We're recruiting a new cardiologist, an interventional cardiologist that does translational research. He's going to use our biorepository. It's open for the Brain Center to put samples in there if they so wish. And Ellen has really pioneered the quality control in the RNA and the DNA testing. We've been doing that for other biorepositories around the region. We're sort of their we do their quality control for them. Because we were the first ones that began doing that, realized the importance of assuring that what we've put away has good RNA, good DNA. We also beat all of us to the punch because of the last 4 years, we've been collecting blood samples on normal patients through our Partners in Wellness Cancer Screening Program. The van you saw, we go out and screen about 5,000 patients in North and Central Louisiana, providing free cancer screening services for those that can't afford it. Or those that have insurance, we now do commercial testing. We provide easy access to service. You know, many of our rural patients live 100 miles away from a mammogram machine. It's very hard if you have limited income to get to that center. But we bring the mammogram machine to you to get your screening. And so those people have been asked to participate in giving us blood samples. So we're building up a biorepository of normal patients where if they develop cancer in the future, we can go back and look for biomarkers. And in the genomic sequencing lab, this is open for the residents and students. If you want to be involved in research, Dr. Summerhalder, one of our residents who graduated last year, just joined us in oncology, just spent a month in the lab learning how to do genomic sequencing and has an IRB-approved protocol looking in breast cancer at the genomics of women before and after neoadjuvant chemotherapy. So this is something if you want to be involved in, we're more than happy to bring you on board, and Ellen and Adam Greer to teach you about the sequencing and help you do it. It's a resource for education. And I think for those of you that might want to pursue a research career in the future, we would really like to work with you and give you a good start, if that's what you so wish. And then I thought we would have Mindie is going to sort of talk to you about how we're using this information now in the clinic to help our patients.
Let me ask you a question before Mindie speaks. Where do you think, I was thinking that you guys are lucky because you're in, as this is taking off. If it's 5 years from now, where do you envision what y'all are talking about at LSU and the Ochsner deal? And
Oh, I think we're going to have a statewide network. We're already talking about it. We're talking about it in oncology. I'm sure the brain people are talking about it in the cardiovascular. We want to bring and pool all of our data together as a state so that we've got a tremendous database through the LCATS big data project. That's what we're looking at doing. And I think it's going to get even more fascinating. What we're looking at now is circulating tumor DNA or circulating DNA. You know, when your cells die, you got to get rid of the waste products through the kidney, or the liver. So the DNA is in the blood. So we can don't just have to biopsy a tumor, we can actually pull a blood sample and find small DNA fragments in it and do sequencing from the blood. And then as the DNA gets metabolized further, it's excreted in the urine. That's why all of us does a urine. You can find the DNA going out in the urine. You can do your complete sequencing from blood or urine. You don't even need to do tumor biopsies anymore. It's a little more tedious and it's a little more involved, but we have a big project under Adam Greer looking at circulating tumor DNA changes in patients as they receive their chemotherapy for prognosis, or also to look for evolution of their cancer and the development of new clones of cancer with different DNA patterns. So this is a huge technology. It's just, it's mind-boggling when I look at this. I wish my career was about 20 years to go in it rather than maybe 10 years at the best. It's going to be fascinating I think a lot of what we do, not just in oncology, but in all the specialties of medicine, are going to be genomic based. And people are going to come in, give their DNA, and you're going to know what diseases they're at risk for, what things you need to worry about. And then maybe you can really start talking about prevention. If you know somebody's got a bad set of genes that's going to promote atherosclerotic disease and lipid disorders, you can start intervening much earlier than waiting until they come in with their myocardial infarction and have to have their stent done. So I think it's going to lead to a world of prevention, which is much better than the world of treatment that we live in right now.
So I wanted Mindie is also one of my favorite student resident fellow faculty. And I think one of the reasons I want you to hear from her is, does this help her? Does this help her patients? I believe that all of y'all will be involved in research, whether you're in the private sector or academia. It is just, as you've heard Dr. Mills, it's going to be the way it is. So Mindie, how is all this helping your patients, your fellows, and how does it help you?
So there's a lot of different ways that I use it for patients currently. I can give you kind of two examples. Normally in cancer, the way we treat patients is you have colon cancer, you're pretty much going to get this set of medications. And we know that because patients had a biopsy, they come to me and the biopsy says colon cancer. Well, things are changing now, and colon cancer isn't treated as colon cancer. It's treated as colon cancer that has this mutation, it has this change, and it has this change, which may mean that the three of us sitting up here all have colon cancer, but we all may need a separate drug right from the start. So being able to have the Cancer Plex test on our campus and so easily available allows me to order the test up front, and when a patient comes to see me, instead of saying, well, we have colon cancer, we know you have colon cancer, but I can't really tell you what we're going to do because I don't have all the information I need right now. I can be equipped with all the information I need to talk to a patient about why they may get different medications, because people talk and people assume that, okay, you had colon cancer and I have colon cancer, so we're going to have the exact same side effects, exact same things that happen, but we know that doesn't happen. And then the patients hear stories about, oh, well, I don't want that medicine, or I don't want that chemotherapy because it was terrible. So now when a patient comes to me and I have all the information I need to know, do they need medicine A, B, and C, or do they need medicine A, B, and D, I can talk to the patients about it right up front and let them know exactly why. And patients are happy to know that we're treating them based on their cancer specifically, not just based on the fact that they have colon cancer like everybody else. Because patients do, and it's not just educated, higher education patients. Patients have heard about precision medicine and specific medications treating different cancers and targeting different things. So it's nice when they come in and I can say, yeah, you're right, that your cancer has this and it has this, which means we can use certain medications. And when I say certain medications, we saw on the report, if a patient has, say, an EGFR mutation, in colon cancer, there's a medicine that I know I shouldn't use. It's not going to work. Right? If I don't have that information, I don't know if I should use that medicine or not. And as you guys probably know, a lot of chemotherapies aren't just a medication you want to randomly give to somebody. So by having the Cancer Plex test, I can get all this information quickly, and I can say, well, you're not really going to benefit from this medication, so I'm not going to put you through the side effects. Another way I use the Cancer Plex test, besides just knowing all the information up front, is I have a lot of patients who, we try the traditional chemotherapies that we have always thought were the best, and for some reason the cancer doesn't respond at all. Okay, well, it's colon cancer, it's supposed to respond to these medicines, but it doesn't respond at all. And then you try to do what we would consider the best second-line treatment. Well, it still doesn't respond. Okay, you now have a very healthy 40-year-old person that you're looking in the face saying, I can't do anything else for you. And I don't understand why your cancer didn't get better with these medicines. Having the Cancerplex test, the first time I realized that maybe this is a little bit more aggressive, or for some reason they're not responding, I can go ahead and order this test, and it will tell me maybe there's some different mutations that make that we're starting to learn now are more aggressive type of colon cancer. Or maybe there's some mutations that in other cancers we know respond to certain drugs that maybe we can try because this patient's healthy. And, you know, it's not worth just saying, sorry, I can't do anything else for you. And the good thing about it is that it's very easy to read. You don't have to be a cancer doctor to look at these reports and to understand them. I show these reports to my patients, and I explain to them. Now, do they understand all the words and what the mutations mean? No, but that's not really the important thing about it. It's just kind of showing them the general areas, and this is what I'm looking at, and this is what your tumor has, and why we think your tumor is different than someone else with colon cancer. I had a patient recently. She was a lady, I think she was 56, had been being treated for about 6 years or so with metastatic colon cancer. So her tumor was not extremely aggressive, but she was still very healthy, working, had I think 2 grandkids, 1 on the way, and then the chemotherapy that we were using stopped working. So I had ordered Cancerplex testing, and it came back with a mutation and 2 medications that were used in a different type of cancer, but were used because of this specific mutation. So we talked to her about it. I explained to her that that this hasn't been studied officially in colon cancer, but I also explained that we're moving away from kind of that using cancer type to treat patients, and that other types of cancers had worked with this mutation. So she tried it, and now has been using that medication. It's only been a month or so, but she's doing really well, no side effects. So I'm hoping that means maybe she's responding, because she was kind of starting to lose weight pretty quickly and getting a little more fatigued, and now she's doing better, back to working, things like that. So my hopes are that that will work. It won't work forever, but at least maybe for the foreseeable future. Without having the Cancerplex test, I wouldn't have been able to do that, because I can't just give someone pick a completely random medicine out of the air and say, here you go, try this one. So without having that, she would have ended up on hospice and not having anything else to try. So that is a big thing too, is being able to try treatments that we know work in other cancers with the same mutation. And like we're kind of talking about, I think that's how cancer treatment is going to be moving. I don't think we're going to be treating colon cancer like colon cancer in the future.
Yeah, if you look at what's happened in oncology, it's going to be happening in everything. 95% of the new drugs approved in the last 3 years are targeted therapy based on mutations, and not the classic chemotherapy drugs. We're not going to see classic chemotherapy drugs approved by the FDA anymore. And in fact, 2 months ago, the FDA approved the first chemo drug based on the genomic profile and not the histology. So you could have breast, lung, colon, prostate. If you have that genetic profile, that drug's been approved by the FDA. And it's a first ever, and I think you're going to see a lot more of this, that drugs are going to be approved by the FDA based on their genomics. And it's going to be happening in neuro, it's going to be happening across the board. The number 2 user of genomic testing right now, you would never believe, the number 2 specialty that uses this, not cancer, it's neonatology and prenatal care. It's looking at the children for genomic defects. And in fact, you can check the mother's blood, not the baby, when they're pregnant, and find defects in the fetus. Because that DNA's got to get out some way, and it goes right out through the mother's bloodstream. And this gives tremendous importance for the high-risk OB and the perinatal, taking care of the baby in utero. We can extend our care of the child now to help correct genetic or have the baby born normally. You can do that in a disease like severe beta thalassemia. If you detect it early enough, you can give blood transfusions in utero, and the babies are born normal. So there's all sorts of possibilities opening up with the genomic testing that you wouldn't, normally think about. I think it's a whole new arena for us.
So using it for our fellows is one thing that we kind of talk about quickly. For our fellows, it's just an educational opportunity that not a lot of even oncology fellows get exposed to. And this is something that they are going to be relying on as soon as they get into practice. And as fast as medicine is moving now, it's very hard to keep up being in private practice or even in small academic centers. It's hard to keep up with these things. So the fact that our fellows have this, just right here, as well as being able to use it for research, gives them a, kind of a big head start as they go into their careers. A fellow that graduated recently was talking about they were taking the next-gen sequencing testing into their entire group because their group didn't know anything about it, and so they were very resistant to use it because it is a learning process. So we're hoping also that our fellows will be able to take it as they move on with their careers into their communities as well.
Oh, it's clear we need to do a better job of education in our state of our practitioners that are using this. If I started talking to groups around the state about this, it's perfectly clear that many of the practices are behind the times, and we've got to bring them up to the more modern times.
So, what's the bottom line for LSU, for Louisiana, for all of us in this room? I think Bob Willem said it this. They are changing.
There you go, Jase. You worked it in.
Gather around people wherever you roam and admit that the waters around you have grown, and accept that soon you'll be drenched to the bone. If your time to you is worth saving, then you better start swimming or you'll sink like a stone. So LSU and Louisiana, the times they are changing. So thank you all for coming. I appreciate that.